Hi, it's great to be speaking with everyone this afternoon. My name is Kevin McDonald, and I'm a new lead fisheries biologist out of the Alpena Fish and Wildlife Conservation Office. This is my first Great Lakes Fisheries Commission annual meeting, and I'm really excited to share some of the work we've been up to regarding Cisco rehabilitation in Lake Huron. In particular, I'll be sharing an update on a multi-agency effort to restore Cisco populations in Saginaw Bay. I need to mention before moving forward that a lot of what I'll be sharing today is the result of a lot of collaboration between several partners and just wouldn't be possible without the input of everyone involved. So this afternoon I'll be focusing in on our efforts in Saginaw Bay. So beginning in 2018 and continuing to present, the U.S. Fish and Wildlife Service began stocking Saginaw Bay with Cisco. Those fish were derived from a broodstock line collected from northern Lake Huron, specifically the Lake Huron and Drummond Island areas. And currently those lines of broodstock were both developed and reared at Jordan River National Fish Hatchery. And we are now far enough along into this effort that we can start to evaluate how successful those stocking events actually are or were. So today I'll be sharing some information on how we anticipate to evaluate this population and some of the tools that we've been using and plan on using in the future. So the first topic that I'm going to cover is just a simple update on our the stocking and gamete collections that we did in 2020. So in 2020, we were able to stock just under 200,000 Cisco in the spring and just over 470,000 Cisco in the fall. So in total, it's about 672,000 Cisco stocked in the Saginaw Bay for 2020. You may notice that number, 672,000 fish, is quite a bit lower than our original goal of 1.1 million. We unfortunately had a water temperature issue at the hatchery this year and lost a significant portion of our eggs. So that is why that number is quite a bit lower. In the fall, we were able to get up to Drummond Island and collect 111 pairs of Cisco and bring them back to Jordan River. And we now have enough broodstock at Jordan River where in 2021 we anticipate being able to rely on our hatchery broodstock and will not need to do gamete collections, which is great news. Okay, now I'd like to discuss just quickly one of the tools we have to evaluate survival of these hatchery fish, or these stocked fish, which is oxytetracycline, or OTC. And just real briefly, OTC, it's a chemical that can be introduced into the feed of hatchery fish and actually lays down a mark on hard structures in the fish that then fluoresces under UV light. So it acts as an internal mark for fish that allows us to then, let us know that it's hatchery fish. And so it's a great tool for marking fish that are potentially too small for traditional marks such as pit tags or floy tags or what have you. So both the Alpena Fish and Wildlife Service and Alpena Michigan DNR offices are both fully equipped to examine Cisco structures, looking for the presence or absence of the OTC mark to be able to differentiate hatchery and wild Cisco that will start coming into our surveys. And we've been working to set standards to QA/QC this process because how you prepare these structures can affect your ability to read an OTC mark and affect accuracy. So we actually developed an SOP, a standard operating procedure for OTC extraction and examination of the structures. And one of the things that came out of that was we did some time trials to look at the length of time that it takes under the fluorescent light in order for the marks to start to fluoresce. And what we found was it could take upwards of 10 minutes to get these OTC marks to fluoresce in a reliable manner. And so on this slide, we can see this is the same structure under the initial exposure, 3 minutes, 6 minutes, and 10 minutes. And you can tell if you look at the initial exposure, you might miss one of those rings, and it does take quite a bit. So that's something that we're taking to heart, and as we've incorporated in our SOP to increase our accuracy of our reads. And something that I'm real happy that we were able to discover and figure out. One of the cool things with OTC is our ability to do differential marks between our spring release and fall releases. So for these fish, the spring release Cisco, the smaller fish, receive a single OTC treatment and thus have a single mark. And our fall fish receive 2 OTC treatments, and then their hard structures actually have 2 rings So that allows us, when we do recapture these fish, to be able to associate them back to their release season. So as I mentioned, our spring fish, they receive a single mark. These were really difficult structures to examine just because they are on such small fish. And just extracting these vertebrae becomes more art than science. But as you can see, it can be done with these tiny fish, and you get some really clear marks. And so if we contrast that with, and so here's an example of a fall fish. So as I mentioned, fall fish receive 2 OTC treatments, so they have 2 marks. These marks should be evident on kind of either side of these spine portions of the vertebrae. And and so these fish are a little easier to read just because they end up being a bit bigger in size and a little less delicate. And I should also mention that the reason we're using vertebrae is they seem to be a little more robust structure when it comes to little, especially little guys. And so we end up using those rather than otoliths or maxilla. They just end up being a little bit more reliable. And the other thing I'd like to mention is, so in 2018, all the fish were fall fish, or released in the fall, and received a double mark at OTC. And so that year class, there was no spring fish. So if we do end up, if those fish do recruit to our gear, we end up catching them in numbers, we'll have a nice little natural experiment to assess how well the double mark wears in the lake. And that should really kind of help our precision and our accuracy of assessing, a true double mark versus a true single mark, and that we're not missing marks. So I'm looking forward to that, and hopefully we collect a bunch of those fish. So moving on, next thing I'd like to discuss is just how exactly we're planning on catching these fish, and the development of some targeted surveys in Saginaw Bay looking for these stocked Cisco. So one of the tools that we'll be using to hopefully sample this population of stocked Cisco is targeted midwater trawls. So beginning in 2004, USGS started doing acoustic midwater trawls, and so Fish and Wildlife Service and USGS were able to kind of leverage these surveys to help assess or evaluate the post-stocking survival of these Cisco. And then kind of within Saginaw Bay and the inner bay there, Michigan DNR will be operating some midwater trawls to hopefully look for fish, closer or further in the bay, I should say. The other surveys that will be kind of new surveys, I should say, that we'll be implementing is we'll be looking, Fish and Wildlife Service, so my crew will be doing some fall adult spawning surveys. And so these are brand new, and so they're pretty exploratory because we're uncertain if these fish are going to be coming back right to where they were stocked originally. As I mentioned, the first year class was stocked in 2018, so this year would kind of be the first year that we would expect to see spawning adults. And so we're going to be implementing some small mesh gillnet. The plan is to kind of start our survey around where they originally stocked there on the map. And and then hopefully if we don't find them, we'll kind of expand our search grid and kind of take it from there. Or, I start targeting other habitats, other spawning areas that, or areas I should say, that have spawning habitat nearby. But hopefully we'll find them and be able to get on them and sample some of these fish. So the other part of this project is, or I should say that this effort is, the existing surveys. What else have we been doing to assess Cisco, or the status of Cisco in Saginaw Bay? So 2 surveys that Michigan DNR will be doing this coming year is their Saginaw Bay Fish Community Survey and their Spring Lake Trout surveys. So in the community survey, that's a gill netting and bottom trawling within Saginaw Bay. And then in the spring, the lake trout surveys, they do gill nets and we'll also be examining the stomach contents of the predators caught in that survey to look for Cisco. So hopefully, we'll be finding fish in those surveys as well. So a lot of all the other surveys that I've discussed so far have been, aimed at finding adult fish. adult Cisco. And so some surveys that my office has been undertaking has been early life history to document if any wild recruitment has been occurring. And so these surveys have become kind of even more important as we expect those stocked fish to reach maturity and begin spawning potentially. So yeah, so Fish and Wildlife Service will be doing continue our early life history surveys in Saginaw Bay. And so to do that, we do two types of surveys. The first is doing Newston netting, so ichthyoplankton tows, looking for larval fish, kind of in more pelagic habitats there, and all that sampling is done, depth stratified. And the other type of surveys that we do is beach seining, looking for more juvenile fish in more littoral habitats close to shore. And then also we do some small boat bottom trawling a bit closer to shore too as well, looking for those juvenile fish. So hopefully if these fish do start recruiting and start spawning, we'll be able to catch them at that phase of their life as well. And so the last survey that I wanted to discuss is the bottom trawl survey led by USGS. And so this figure kind of shows all the different potential sampling locations. I believe there's a total of 131 of them. Where they're able to sample in 2019, and then where those are in relation to the Cisco stocking locations in Saginaw Bay. And so the hope here is that if stocked fish, or hatchery fish, I should say, moved into the outer bay, closer to the main basin, hopefully they'll show up in these surveys. And so, yeah, these are deeper surveys between 8 and 65 meters, and are going to cover a variety of bottom habitats, including sand, silt, and gravel. And they'll all be done out of Tawas there. And so I hope, I painted a picture here that shows all the variety of methods we're going to use to try to, sample and evaluate this population, ranging from, the early life history all the way to adult spawning surveys and everything in between. So we're really excited to move forward with the sampling, especially as I mentioned, these fish should be recruiting to our gear starting this year. And so we're really excited to see what happens, and hopefully we're going to catch some fish. And so the last thing that I want to talk about, kind of an important component of this project, has been our community outreach, and then also the partnerships that have made it possible. So in terms of community outreach, we've been able to do several presentations about this rehabilitation project, both at the International Association of Great Lakes Research and also at Lake Huron Fisheries Advisory Council meetings. And another tool that we've been able to put out there in conjunction with the Michigan Sea Grant has been these Cisco wanted posters that have gone up at boat launches and places where there's recreational angling. Hopefully I didn't, inform the public that if they catch a Cisco, especially in Saginaw Bay, that we're able to collect samples from that and be able to kind of use the public as a means to sample fish for us. And so we're really excited, and hopefully we'll be able to get some fish in that manner as well. And so the last thing I wanted to mention is just all the partnerships that have been integral to making this effort, work. And so the first one is, we've been working with MSU to collect our unidentifiable Corrigonans from our predator diets in Saginaw Bay, and they've been helping us with that effort. The Michigan DNR Creel program and also the Fish and Wildlife Service mass marking program also been a really big help helping us with these evaluations. Michigan Sea Grant and MSU Extension Office, we've been working with them, as I mentioned, with the poster, the Cisco Wanted poster, and also just other outreach events. NOAA, the Gloral Lab, has been phenomenal, helping us get our diet analysis and zooplankton done from our early life history surveys. And then also GLaDOS, just helping with doing movement and providing those resources to do any telemetry research that is coming down the pipeline. And so I hope as you can tell just from the bottom half of this slide, this has been a big project with a lot of people contributing, and it just wouldn't be possible without them. So I'd like to thank all of our partners, and with that, I believe I can take some questions. Thank you.